Hi again, everybody. So I had some questions that were kind of presented about the glow-in-the-dark rats that you guys saw on the first page of the module. So I thought I'd give you a quick explanation of those glow-in-the-dark rats. But the explanation's kind of lengthy and really requires somebody to actually sit down and explain it. So I thought I'd make you a video today of how that works. And it starts with a process called recombinant DNA technology. It's also often called transformation because you're actually transforming the genes of an organism. And basically this is a procedure for moving a gene from one organism into another organism. And this is true, live, honest-to-God genetic engineering. This is how genetic engineering is accomplished, because we're actually putting genes from one organism into somebody else. It starts with restriction enzymes, and restriction enzymes are special enzymes that are going to cut the DNA at very specific sequences. So they recognize a very specific sequence, and you can see in the picture here, this EcoR1 is a restriction enzyme, and it cuts at AATT. So essentially what you're seeing is a picture of a restriction enzyme at work. Now, I should mention before we begin that DNA in bacteria is kind of weird. The bacteria has its own chromosome, and that's where most of its genes are located, but it also has these little plasmids, which are little circular pieces of DNA. So what you're seeing here is one of these plasmids taken out of the bacterium and being cut with EcoR1, which is a restriction enzyme. Now, we use the same restriction enzyme to take a gene from another organism. Now, this is commonly done to try to engineer bacteria to produce helpful things to us. For example, human insulin. Diabetics need to have insulin. You can't always use slaughterhouse insulin that you'd get from cows or pigs or things like that at a slaughterhouse, because the animal insulin can be bad for a person's system. You can get immune to it, you can have a tissue rejection reaction, and so on. So the way that scientists make insulin is by taking the human insulin gene and cutting it out of a human chromosome, which is what you're seeing right here. This is a human gene being cut out. They open up one of these plasmids, which is what you're seeing right here. That's the plasmid opened up. And then they stick the gene and the plasmid together to get this resultant plasmid that you see here. So you've got the purple gene that you've got inserted into the DNA of another organism. That's genetic engineering. That's recombinant DNA, because you're combining DNA from two different sources. The next step here is to then take our plasmid, which you see in this picture in the top corner here, and insert it into a bacterial cell. And now this bacterial cell is transformed. We call it a genetically modified organism. It's sometimes also called a transgenic bacterium. Now here's the thing. It's all well and good to see all these pictures and to see mechanically how this works, but remember we're talking about a microorganism. And remember that you're talking about DNA, which is also very, very microscopic. So what this looks like in a lab is what you see in this picture right here. This is a centrifuge tube, and this little pellet right here in the bottom of the tube, that's the bacteria that's been transformed. And just by looking at that pellet, you can't tell if this actually worked. You know, we've dumped our bacteria into that tube, we've dumped the plasmid into that tube, and we hope the bacteria have picked up the plasmid, but we don't really know. So we need a way to test to see if the bacteria have actually picked up the plasmid and will therefore produce insulin for us. So what scientists have done is come up with a procedure to insert a test gene. So back up to our plasmid here. This little plasmid right here often doesn't just contain a human insulin gene or whatever gene we're trying to get into the plasmid. It's also going to contain a gene for resistance to the antibiotic ampicillin. So this is a type of penicillin. You might have had this if you've had strep throat or something like that. But ampicillin is an antibiotic, which means it normally kills bacteria. So we're actually engineering our bacteria to be resistant to this ampicillin. Okay, so now if we take our bacteria here, our transformed bacteria, and we grow it up on a plate, and we put ampicillin on our plate, only bacteria that have picked up our plasmid, only true genetically engineered bacteria, will be able to grow on our plate here. So if the bacteria grows on our plate, we know that the bacteria has picked up the plasmid. We know then if they've picked up the plasmid that that plasmid also contains the gene that we want them to have. So there's a test procedure in place. This is the old school test procedure. There are some new school test procedures, and this is where we start to circle back to our glow-in-the-dark rats. Scientists don't necessarily like the idea of engineering bacteria that are antibiotic resistant for some pretty obvious reasons. If these antibiotic resistant bacteria were to get out, they could make you very sick and then they wouldn't respond to normal antibiotics, so that would be a very bad thing. Scientists have switched tactics then, and they've gone to inserting genes for fluorescence. So fluorescence means if I strike an object that's fluorescent with UV light, like a black light, it emits a glow or light. And so scientists have started, instead of using antibiotics or antibiotic resistance, they've started inserting green and blue and red fluorescent genes into these bacteria. That way, when they grow the bacteria on a plate, if they shine a black light on the bacteria and the bacteria glows the color that they want it to, then they know that that gene, which was tied to another gene they inserted, is also there. All right, now, here's the thing, too. This same procedure is the first step in all genetic engineering. So far we've only talked about bacteria. Don't forget, we've discussed that there are other players in biotechnology. There's not just bacteria, there's also funguses. We can engineer yeast. We can engineer viruses as well. Okay, so while we do a lot of genetic engineering with bacteria, you can also do this with funguses and viruses. And that's going to come into play here in just a minute, because for this kind of genetic engineering, we need the bacteria, the yeast, and the fungus, and the viruses are all really easy to manipulate. They're all single cellular, or a virus is not made of cells at all. And the thing that makes bacteria and viruses superior for use in this kind of genetic engineering is that they're good at getting inside of cells. In other words, they can cause infections. So what you're seeing is how we take that procedure and we turn this genetically engineered organism right here into a vector for moving that gene now into another organism. So what you're seeing is an agrobacterium. An agrobacterium is very popular for use in genetically engineering or transforming plants. So the cell that you see here is a plant cell. And what we're doing is we're allowing this agrobacterium that now contains a plasmid that we want to infect our plant cell. We can then take, and this picture up here at the top also has the same procedure. It's got a couple extra steps, but basically here's our agrobacterium, and we insert that agrobacterium into these plant cells, and we let the plant cells grow up. And once the plant cells have been infected with that agrobacterium, they now contain the gene that the agrobacterium brought inside. And so now that plant cell has whatever the engineered gene is. So this is how golden rice was made. You've heard about golden rice in one of our learning blocks. This is how you get BT crops and stuff like that. So when we talk about genetically engineering crops, that's what we're talking about. We take our recombinant plasmid inside of a bacterium and we let that bacterium infect a plant cell. This also, though, works with animals. So you can actually do the same thing, the same procedure, except that with animals they usually engineer a virus as the vector. So instead of an agrobacterium that you see here, there'd be some sort of virus that they would engineer to move the gene over. Now for the reason you watched this whole presentation. All right, so let's back up now to the glow-in-the-dark rats. The glow-in-the-dark rats are the result of a jellyfish gene for fluorescence. Remember that term fluorescence, you've seen it before, being inserted into a rat, okay? And then the result then is once that gene goes into the rat, you have these little babies that when you shine a light on them, they fluoresce, they glow under the UV light. So they glow green or they glow purple or whatever the chemical they're producing is. All right, remember what I said. You cannot see a gene entering a cell. Because you can't see that, you can't be sure that your procedure worked. You can't be 100% certain that when the bacteria was inserted into the organism to cause the infection, to transform the organism, or when the virus was inserted into the organism, you can't be sure that the virus actually got into the organism's cells. So, we focus on glow in the dark a lot because it's cool. We inserted a jellyfish gene into a rat or into rabbits. But the real reason for that glow in the dark gene is that that gene acts just like our antibiotic resistance did in the bacteria. When scientists in a lab want to engineer rats or rabbits to have Alzheimer's or to be obese or things like that so that they can study the progression of diseases and test treatments on those animals before they test them in humans, they need to make sure that those animals are going to actually have the genes that they're trying to engineer. And so they insert the fluorescence gene in the animal's genome by using that same vector, and they know then if they have these little baby rats and they shine some light on the baby rats and the rats fluoresce, then scientists know that the transformation worked and that these baby rats are now going to have the genes that they wanted them to have, and they can do their experiments on them. So they can watch how things like diabetes develops in humans by looking at it in the rat. They can look at how Alzheimer's develops by looking at it in the rat. And then they can study how the disease progresses and how to treat it. So, believe it or not, there's actually a reason for glow-in-the-dark rats beyond the fact that it's just pretty cool.